Hello and welcome back to part 2 of our look at the introduction into anatomy and organization of the nervous system. And this is the slide where we left off at the end of the last video, kind of reviewing neural anatomy. In other words, what is the anatomy of a neuron? Which kind of an overlap with lab, but I wanted to spend at least a few seconds just kind of reviewing that, making sure that you guys were good with that. And the focus of this video will be the myelin sheath and exactly what is or is not myelin. And so we know that both within the central nervous system and the peripheral nervous system, that myelin is present. It wraps around the axon. It acts as a protective barrier for that axon. We know that within the central nervous system, that myelin is formed from these extensions coming off of the cell body of an oligodendrocyte. Um, and within the peripheral nervous system, we know that the myelin sheath is created from Schwann cells or neurolemmacytes that completely wrap themselves around the axon. But that is all we know from the video that in the lecture that we've had so far. And so I wanted to spend a little bit more time just kind of talking about myelin and some of the differences again with myelin between the peripheral nervous system and the central nervous system. And so to start off, first of all, what is myelin? And the first thing that we need to keep in mind is that myelin is really a protein lipid. It's a lipoprotein. Um, and the fact that it's made of a lipid means that it's high in fat. And this is why when you look at a cross section of a neuron under the microscope, as we did within the lab, you actually see a ring of white around the axon. That ring of white is indeed the myelin. And it's white because it is high in lipid. Right? Just like adipose tissue is high in lipid and stains usually white, so does the myelin sheath. And so that's the first thing that you really need to keep in mind. And what I've done here is the image that you're looking at really kind of shows, I think, well, um, that analogy that I used, I think, in the last video of myelin and the myelin sheath in conjunction with the axon being very analogous to that of an electrical wire or an extension cord. Right? Whereas the wires within the extension cord are indeed the axon that it's coming off of the cell body of the neuron. And so this axon is a collection of nerves, a collection of fibers that are able to transmit an electrical impulse, aka an action potential. Right? Just like the wires running through a cord, right, are able to carry the electricity from point A to point B. And what prevents that electricity from leaking out or from shorting within these wires is this rubber insulative covering, right, that is wrapped around the wires, just like an extension cord. That orange covering around the extension cord. Well, uh, our axons need that same kind of protection. And so what we see is we see the myelin that is wrapped around the axon is really serving as that outer exterior of an extension cord that prevents the electrical charge, the electricity, the action potential from escaping, which allows it to make sure that it in a very rapid fashion, get from point A to point B. And so myelin has this kind of love-hate relationship in that it's doing two things. One, myelin is reducing ion leakage. Right? And specifically, the ion that is being targeted is sodium ions. Right? Sodium ions. And so it's reducing that leakage, and as you reduce the leakage of sodium, you're actually increasing the speed at which the action potential is moving. But on the flip side of that, because the sodium is not able to escape, it's actually rubbing up against the myelin itself. And so we are actually increasing resistance. And because we are increasing resistance, we actually lose some speed, which is why those nodes of Ranvier are so important. And this is where the nodes of Ranvier come into play, because they are unmyelinated. The nodes of Ranvier lack the myelin, which means they have sodium channels that are allowing the sodium to be able to freely move, repropagating the action potential. And so that is really, really important. The other thing to keep in mind here is not all axons are myelinated. Right? We see non-myelinated fibers in neural pathways, especially within the postganglion neurons. That's going to make more sense once we get to the autonomic nervous system. We see non-myelinated fibers where we've got very short distances that action potentials are traveling. So think about the back of the eye with the retina. You have axons, you have neural cells, nerve cells that are making up that retinal layer to transmit light energy and convert it over into light stimulus, electrical impulses that the brain can then interpret as images. Well, it's such a short distance between the back of the eye and the optic nerve that those neurons, those axons, are not myelinated, and we see that also within the optic tract. We see that also within the mucous membrane within the nose and the olfactory nerves. They are free ends. In other words, they lack myelination, and so while they conduct more slowly because of the loss of ions, right, ion leakage, the fact that they're going such a short distance doesn't really hold anything down. Right? And so we do see instances of non-myelinated fibers. By the way, it's the loss of myelin. It's when myelinated axons become non-myelinated that we have issues. Right? So think of things like dementia and Alzheimer's and ALS or Lou Gehrig's disease. A lot of those types of diseases are because of a lack of myelin, and so myelin loss. And when you lose that myelin, those axons become leaky, and sometimes the action potential, the stimulus, never reaches its intended target, and so it definitely does slow it down over a larger distance. So keep that in mind. Right? Don't lose sight of that. As we have talked about in the previous video, within the peripheral nervous system, I'm going to start with the peripheral nervous system because I think this is a little bit more impactful than maybe starting with the central nervous system. And I'm sorry, I get tired of using the same laser color. Let's go with blue. Okay. So again, here's the axon. All right, so you got to kind of remember down here, there's a cell body, there's some dendrites over there, but this here's the axon. All right, and you can see here that here is a Schwann cell. All right, And this Schwann cell begins to hug the axon. And as it does that, again, pay attention, here's the nucleus over here, right? As it does that, this cell body begins to wrap and wrap and wrap and wrap. I want to point out and stress the fact that the nucleus of the neurolemmacyte, or the Schwann cell, remains outside. It remains superficial. And so it's the rest of the Schwann cell that actually wraps and wraps and wraps. And you can see here we've got more wrapping. You can see here you've got more wrapping. Right? 60 to 80 times that cell is going to wrap around. And of course, every time it does that, it gets a little bit thinner. Right? Every time it does that, that cell itself gets a little bit thinner. Now, once it is done wrapping, right, you've got a little bit of a bulge that is on the superficial edge of the newly created segment of myelin. All right, and that bulge is where the nucleus is of the Schwann cell. Right? And that bulge, and which again, remember, contains not only the nucleus, but the majority of the cytoplasm, because remember, the cell membrane, the cell here, has gotten thinner and thinner and thinner each time it wraps, right? And that cytoplasm had to go someplace. So where did it go? It collected itself. It accumulated here within this peripheral bulge that contains the nucleus. Well, that bulge that contains the nucleus and the majority of the cytoplasm, or the cytosol, is what we call the neurolemma. Right? Which is why we refer to this as a neurolemmacyte. Right? Schwann cells are also referred to as a neurolemmacyte. Right? Let me spell that out for you guys since it's not on there. Neurolemmacyte. All right, now you will also see neurolemma spelled neurolemma. Either spelling is correct. Right? Either spelling is correct. I know you guys hate that, but it's true. All right. And so the fact that the reason why we call this a neurolemmacyte is because it contains a neurolemma. It contains this peripheral bulge with the nucleus and the majority of the cytoplasm that's contained from within the Schwann cell, or the neurolemmacyte. The other interesting part about this is the Schwann cell here contains no sodium channels, no potassium channels, has no protein carriers in it whatsoever, and so this makes a really good insulator. This is why we use this as a conduit, if you would, for myelin. Now, let's contrast that. Let's contrast that with what we see happening within the central nervous system, because it's a little bit different of a story that we see within the central nervous system. I mean, what we end up seeing with the central nervous system here is, oh good, it's still in blue. We have these oligodendrocytes. Right? We have these oligodendrocytes whose cell body, and therefore nucleus, remains independent of the myelin that is being created by these extensions. And so the oligodendrocyte will go ahead and create this arm that reaches out, grabs onto, and begins to wrap itself around a segment of axon. Now here's the interesting thing. Each oligodendrocyte has the ability to create up to about 60 or so independent segments of myelin. Now that might seem like a lot, but it's not, considering from a cellular perspective that most things function within the thousands to tens of thousands, upwards to the millions. The fact that each of these oligodendrocytes can only produce 60 branches or 60 offshoots is minimal, which is why oligo here, if you look up the word oligo, it means few. Right? Dendro is referring to tree branches or branching of a tree. And so these are cells that create few branches, even though, again, 60 might seem like a lot. It's not. All right? But there is no neurolemma that's associated with the myelin here. And that's important. That is extremely important when we consider nerve regeneration. That's going to be the difference. And we're going to touch on that here in a few minutes. Right, we're going to touch on that here in a few minutes. All right. So again, what makes the oligodendrocytes special, what makes the myelin special within the central nervous system, is the fact that there is no neurolemma that is present within the myelin sheath, because the nucleus in the cell body remains independent. Right? It remains independent. And so, with all of that said, how does this apply to nerve repair? Why can we have neural regeneration within the peripheral nervous system, but we don't have the ability, or that ability is severely, severely limited when we look at the central nervous system? Again, part of this has to do with location, location, location. I mean, that's really what it comes down to. And so let's say you've got a peripheral nerve, all right, that is going down the arm, and you go ahead and you're dicing up, I don't know, you're chopping onions or potatoes or tomatoes or cucumber, and the knife slips, and when it slips, you accidentally push the cucumber forward, and the knife goes ahead and cuts through the forearm. Right? Well, when you do that, it's now bleeding, so you've gone deeper than the epidermis. Right? So you've cut through the dermis, you've cut through the subcutaneous level. Maybe you've even gone down into the muscular layer. You know you've cut some nerves. As soon as you enter into the dermis, you know you've cut some nerves. Right? So what happens? Right? How do you not lose feeling or sensation within your lower arm? Why over time do you go ahead and still remain or retain neural feeling? Well, what ends up happening is you create this bulge, all right, around the area that was cut. So here's the nerve. Right? The nerve has been cut, and by saying the nerve has been cut, we mean that the Schwann cell has been compromised and the axon has been compromised as well. Right? That is the first thing. Right? And so what ends up going on here is the axon here creates a plug. Right? And that stops any more action potentials from flowing down here. Action potential cannot get any further than where it is right here. All right, boom, done. All right, that's it. All right, that is all that she wrote on this. All right, so here again you can see you've got your Schwann cells over here. All right, you've got some myelin that has now been released. All right, what we refer to as myelin droplets. All right, and you can see here we've got some axon debris, and we've got this plug that is now right here that is plugging up the axon. It's stopping anything else from moving forward. All right. The next thing that now has to happen is you got all of this damage, and so the next thing that will happen is macrophages are going to come into this area. All right. So look, your Schwann cells right here. Right, your Schwann cells actually create a new barrier that is protective of the cut edge of the axon. And downstream, you've got yourself macrophages. That's what these guys are right here. You've got macrophages that are now going through, and they're cleaning up the debris, the damaged sections of axon, and it's cleaning up the Schwann cells. Right? It's cleaning up the Schwann cells. Well, here's the interesting thing. All right? Here is the interesting things. These, look, here's a neurolemma. Here's a neurolemma. Here's a neurolemma. Here's a neurolemma. These are all of Schwann cells. Here's a neurolemma. Here's a neurolemma. You've got all of these nice little intact Schwann cells that are still present. And what begins to happen is these Schwann cells begin to chemically stimulate the area around it. And that's what you're seeing going on right here. You create something that is referred to as a neural tube, or a neural regeneration tube. And so these growth factors, these interleukins, and you'll learn more about interleukins in AMP2, but these interleukins create this neural tube by which the axon can actually begin to regenerate itself. And it regenerates itself by following that neural tube right on down to where it will reconnect with a healthy part of the axon. And eventually, you'll go ahead and create this new myelin covering, all right, with that is now surrounding the axon. Now, there's varying research out there that says whether or not the node of Ranvier's will actually recreate or regenerate, or whether or not you've got an extended area of myelin, and what that overall impact is on the nervous system. Whatever that is, right, the fact of the matter is you don't lose feeling. You don't lose sensation. You retain the ability to go ahead and not lose the function of everything below the cut. Right? And so that is important. And again, what's driving this is the presence of that neurolemma, right? From wrapping itself around and around and around. That nucleus now has regulation and control that is very localized. And when we look at the central nervous system, the key difference that we see, the reason why you have that lack of ability to drive neural regeneration within the central nervous system is twofold. First thing is, without the neurolemma, you're not instituting, or you're not driving those chemical cues to go ahead and stimulate the formation of the neural tube and stimulation of the new axon. So the lack of a neurolemma is issue number one. Remember, the cell body of the oligodendrocyte remains separate. It doesn't monitor what that myelin is doing. It's just producing it. It doesn't know it's not there anymore because it's in its mind. It's already produced. All right. The other thing is that with the formation of the glial scar, and so there's scarring that happens around that. So that's what you're seeing kind of right here. Right? This is what we would define as glial scarring right there. Right? And you'll notice that it is surrounding the myelin as well. And the fact that that neurolemma site is here and that neurolemma is there, that neurolemma is contained within the glial scarring. Well, there is no neurolemma, once again, within the central nervous system. And so that glial scar actually ends up secreting growth inhibitory substances that will block the oligodendrocytes from even being able to go ahead and access it. Now, it does that in part because it's trying to protect what is still there. Right? So it doesn't want to let anything else in. It completely seals it off. It is a complete roadblock. The other thing that happens is the astrocytes work against this process as well, and the this process is beneficial as a fetus. it's it's not beneficial as an adult, and then the problem is the astrocytes don't lose this ability. They don't differentiate fetal development um, from adult, right, or post fetal development. And what I'm referring to, right, what I'm referring to is something that is referred to as CSPG. All right, and CSPG stands for chondroitin sulfate proteoglycans. All right, chondroitin sulfate, chondroitin sulfate, proteoglycans. Um, and these actually form chains. And these chains attach to the glial scarring and again blocks regeneration. Right? They initiate this release of the or intensify the release of growth inhibitory substances. And so the myelin can't regenerate, the neural tube doesn't regenerate, and therefore the axons themselves also don't regenerate. So it's a small difference. The presence of that neurolemma is a small difference, but it is really the driving force that allows us to have neural regeneration within the peripheral nervous system and inhibits neural regeneration within the central nervous system. And so that's kind of a quick and dirty, down and dirty introduction to what myelin is. Our next video, is going to be focusing on a review of action potentials and really just kind of how does this apply to the nervous system. We know how action potentials are formed and sustained and recreated within the context of the sarcolemma, within specifically skeletal tissue, skeletal muscle tissue. But what does this look like within the nervous system? How similar is it, and what are the peculiarities within the central nervous system that we need to be aware of? And so that's where we're going to be heading in the next video. Take some time, check your notes, compare your notes. Don't forget to look at the mind maps. And hey, guess what? I'll catch you on the flip side.